Welcome everyone to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Shireen Durakshani, and I will be hosting today's session on post-licensing clinical trials for advancing the use of antimicrobials. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial research and development community by facilitating learning, sharing knowledge, and connecting people. The webinars are part of our educational activities. All of our webinars are recorded and are freely available to view after the live broadcast on our website. You can see the link here below. And please don't hesitate to ask questions during the Q&A portion. You can also ask questions during the speakers' presentations. Please make sure to put the name of the speaker your question is intended to. So today's speaker are Stephen Tong and Patrick Harris, and our moderator today is Holly Jackson. Holly got her PhD in statistics and operational research, specializing in adaptive clinical trial designs from Lancaster University in the UK. Her main research interests are novel trial designs, including response adaptive and covariate adjusted response, adaptive randomization designs, sample size re-estimation, and prediction modeling, amongst others. She joined the Infection Prevention and Control research team at the Geneva University Hospitals as a statistician in January 2023. Holly is engaged in ECRADE activities, offering statistical support in the analysis of perpetual observational studies for infectious diseases and the design of embedded trials. She's also involved in research activities to assess the use of novel adaptive trial designs in this setting. Welcome, Holly, and I'm happy to hand over to you to introduce our first speaker.
Hello, everyone. Thanks very much, Shireen. I'm really looking forward to the two presentations we have today in this webinar. Firstly, we have Stephen Tong, who is an infectious disease physician based at the Royal Melbourne Hospital and a professor of infectious diseases at Melbourne Medical School. His research interests include skin pathogens, hospital infections, and influenza, and most recently, he has been involved in two adaptive platform trials. The SNAP trial investigates the treatment of patients with Staph aureus bloodstream infections, and the ASCOT trial looked at the treatment of COVID-19 patients. Links to both of these trials are found at the bottom of the slide. Thanks very much. Stephen, over to you.
Hi there, everyone. Thanks very much, Holly, for that kind introduction. So I've been asked to speak about post-licensing clinical trials for advancing the use of antimicrobials with a focus on the gram-positive side of things, and I think Patrick's going to talk about the gram-negatives. Sorry, there's just a bit of a lag. So my conflicts of interest, I've served on an advisory board for AstraZeneca for a clinical trial for the prevention of Staph aureus bloodstream infections, and I receive royalties from Up to Date for Staph aureus related content pages. Brief outline of my talk. I'm going to focus mainly on two gram-positives, Staphylococcus aureus and Enterococcus, and particularly when they cause bloodstream infections. Speak a little bit about the licensed antibiotics for these two pathogens, what the guidelines say about how we should treat these pathogens, and that really raises some obvious gaps in the knowledge. And then the second half of the talk will be focused mainly on the SNAP trial as a way forward, as an example of an investigator-initiated clinical trial. So in terms of the burden of disease, this is data from the Global Burden of Disease group about the 33 most common bacterial pathogens outside of tuberculosis and the burden that they cause. So the most, I guess, only one that causes an estimated greater than 1 million deaths per year is Staphylococcus aureus. So that's the one on the far left. And of those, about 300,000 are due to bloodstream infections. The next red arrow refers to Enterococcus faecalis and Enterococcus faecium. So I guess if you add those two up together, enterococcal species are probably in the, close to the top 5 of these bacterial pathogens as well. So they do cause a significant and important burden of disease. I've also given some little arrows to other gram-positive organisms, which I really won't talk much about today. Strep pneumoniae, Group B Streptococcus, and Group A Streptococcus as well. So clearly it's not all gram-negatives. Gram-positives form an important burden of disease. And that's also demonstrated in this next slide, which is data from Ontario province in terms of blood culture. So on the x-axis is the positive episodes for a single organism, and on the y-axis is the mortality for each of those positive episodes. So this is province-wide in Ontario in Canada. And you can see that the most common cause of bloodstream infections is E. coli, followed by Staphylococcus aureus. And Enterococcus comes as number 5 on that scale. On the y-axis, though, you can see that something like Staph aureus and Enterococcus have a case fatality rate of somewhere between 20 and 25%. So typically a little bit higher than that due to the gram-negatives. And as a result, in terms of the size of the circles, the largest circle is for Staphylococcus aureus. So of all bloodstream infections, it causes the most deaths, at least in high-income country settings. So what are the licensed antibiotics that we have to treat these infections? So they're actually reasonably limited. For Staph aureus bloodstream infections, we have daptomycin, which was registered and licensed by the FDA in 2006 for bloodstream infections or bacteremia and right-sided infective endocarditis. And then more recently in this past year in 2024, ceftobiprole was given FDA license, again for bacteremia and right-sided infective endocarditis. So surprisingly, there's really only 2 antibiotics which have been licensed for this indication. And then in particular for vancomycin-resistant Enterococcus, there are also 2 drugs, so linezolid for bacteremia, which was provided in 2000, and quinipristin, dalfopristin for E. faecium infections in 1999. And in fact, quinipristin, dalfopristin is no longer available, even though it's licensed, it's no longer available in the United States, the UK or Australia. So if we look at the registrational trials for some of these drugs, so the first one being daptomycin, so there was a publication led by Vance Fowler, published in 2006, which compared daptomycin with the standard therapy at the time. And when you go back to that time, interestingly, gentamicin was included in that standard therapy, which is something that we no longer use now. But they had 246 participants. The primary outcome was treatment success 42 days after the end of therapy. And they used a non-inferiority trial design. So it was non-inferiority with an absolute margin of 20%, assuming a 65% treatment success rate. So it's really, when you think about it, quite a wide margin for non-inferiority. So, allowing down to a 45% treatment success rate if you were to take their initial assumption as being correct. But that's, quite a large non-inferiority margin. And here were their results. This is the forest plot. The black line down the middle is for no difference in the success rates. To the left of that line is where standard therapy is better, and to the right of that line is where daptomycin is better. And they also indicate with the dotted line the minus 20% non-inferiority margin. So if you just look at the top line there with the intention to treat, we can see that it meets that non-inferiority margin. The treatment success was 42.7% with daptomycin and 39.3% with standard therapy. So that's fine. Non-inferiority was met, and there was no difference in mortality either. So 10.8% with daptomycin, 11.3% with standard of care. You could actually make some argument that when looking at new drugs, perhaps we should be seeking superiority, given that a new drug is likely to be more expensive. But nonetheless, they met their non-inferiority margin. Where we're most perhaps interested in using a new drug like daptomycin is perhaps with MRSA, methicillin-resistant Staph aureus infections, rather than MSSA, where we have a plethora of options with beta-lactam antibiotics. And of the MRSA, there were in fact only 89 patients in this trial with MRSA infection. So, when you think about it, this is the main indication that we use this drug for, and there in fact were only 89 patients. In this study itself, in this publication, they don't go on to tell you how many of those patients died. And we had to wait for 2 years in a kind of publication. It's a little bit hidden away. And they showed there that, in fact, the mortality rate, if anything, was higher in the daptomycin group. So 12 out of 45 versus in the standard of care, 8 out of 43 patients. And when we look at the Kaplan-Meier curves, the top line, the black one being the standard of care, vancomycin and gentamicin, and the grey line, the daptomycin group, following patients out to about 150 days, and the probability of survival on the y-axis. You can see that there is actually some separation of the curves in that kind of mid period, and they come together towards the end. And they found that in the log rank test that there was no difference. But note that each of those triangles and each of those squares indicates when a patient was lost to follow-up and therefore censored. So there's actually, for 89 patients altogether, there's a lot of triangles and a lot of squares there. So we're left with, I think, scratching our heads a little bit as to what happened to those patients and truly what was the mortality rate between these two drugs. So my point here is that where we most want to use daptomycin, which is for MRSA infections, there were in fact only 89 patients. This means that there's wide confidence intervals for those estimates of treatment success. And in fact, the mortality was no different between the two drugs, and potentially a bit worse, although I don't think we will ever know that for sure. And subsequently, there's been no clinical trial since 2006, so it's almost 20 years now to address this question, which I think is quite an important question for our field. So the second registrational trial was for ceftobiprole. This was published last year in the New England Journal of Medicine, led by Tom Holland. In this case, they compared ceftobiprole with daptomycin, following on from that previous registrational trial. 390 participants. The primary outcome in this case was overall treatment success, 70 days after randomization. And in this case, the non-inferiority, again, it was non-inferiority design. The margin, absolute margin, was 15%, assuming 40% treatment success. So again, you could argue that, one, should you be making non-inferiority or should it be superiority? And secondly, it's still a reasonably wide non-inferiority margin. Here's their forest plot. Again, non-inferiority was met for all patients. So success rates of 69.8% for ceftobiprole and 68.7% with daptomycin. It wasn't a particularly sick cohort. So out of these 390 patients, there were only 13 deaths in total. And once again, if we look at the MRSA group, which is where, again, we most like to use this drug, I think, there were only 94 patients with MRSA, and thus we have a wide confidence interval for that primary outcome. My point again is where we most want to use ceftobiprole, which I would argue would be for MRSA infections, there were only 94 patients and that wide confidence interval for the estimates of treatment success. And of course, it's been a very recent introduction. I think it's just become available now for broader use, ceftobiprole. There's been no clinical trial since then to address this question. How about linezolid for vancomycin-resistant enterococcus? Well, the screen's blank there because there's been no trials that have led to this registration, and it was based on a large number of observational studies. I'll pull this from the Australian Pharmaceuticals Benefits Advisory Committee meeting from July 2013, and here are the list of studies that the company provided to the PBAC to gain or to seek registration. It would have been very similar data that was used for the FDA registration as well. So we don't even have good clinical trial data to look at the efficacy of linezolid compared to other drugs. There have been 2 trials that have looked at linezolid compared to some other drugs, so tigecycline in one and daptomycin in the other. But you can see that one of them only have 15 patients, and the paper says that's too small appropriately to make any conclusions. And the daptomycin versus linezolid hasn't actually been published, so we don't actually have those data for those 50 patients. But nonetheless, would have been very small and too small to make any firm conclusions. So we have even less data to support use of drugs for the treatment of vancomycin-resistant enterococcus. So based on this, what do our practice guidelines say? And I'm going to focus here on practice guidelines for infective endocarditis because we have several of these, much more so than just for pure bacteremia. AHA stands for American Heart Association, ESC for the European Society of Cardiology, and the Wiki Guidelines is a kind of grassroots, Twitter-inspired group of people who've brought together these guidelines. So one of the points here is that almost none of these recommendations are based on randomized clinical trial data. So if we take the top row, methicillin susceptible Staph aureus, the recommendations are either to use an anti-staphylococcal penicillin, such as nafcillin, oxacillin, or flucloxacillin, or a first-generation cephalosporin in cefazolin. And both the AHA and the ESC guidelines actually say that cefazolin is very much a second-line therapy only if there's an allergy to the first-line therapies. The Wiki Guidelines takes the other approach. They've looked at some observational data that suggests that cefazolin might be a safer and better drug. But there's no randomized clinical trial evidence to support 1 or number 2 for MSSA endocarditis or MSSA bacteremia. And that's just such a key and obvious question that as an infectious diseases clinical trials community, we would like the answer to. And then for MRSA infections, it is vancomycin or daptomycin. And again, as I've shown before, there's only been 89 patients ever randomized into that comparison. Interestingly, the European Society of Cardiology guidelines, they add in all these combinations with other drugs, and again, not supported by any clinical trial evidence. And finally, for prosthetic valve infective endocarditis, the various guidelines suggest the addition of rifampin and gentamicin for AHA and ESC, and for Wiki Guidelines, just the addition of rifampin. And I'll get to showing that there, again, there is very, very little data to support those guideline recommendations. For enterococcal bacteremia, these are the recommendations for Enterococcus faecalis. It's ampicillin or penicillin plus ceftriaxone or gentamicin, and the order of that is different according to AHA or ESC guidelines. And then for VRE, so vancomycin resistance, the recommendations are for linezolid or daptomycin. Both AHA and Wiki Guidelines put them on an equal footing, but once again, there's absolutely no clinical trial evidence to guide what we should do. It might be that one is better than the other, but we just do not know. So if we go back to methicillin susceptible Staph aureus, I've mentioned that the guidelines say either an anti-staphylococcal penicillin or cefazolin. And when a global survey was done asking this question for MSSA bacteremia, what is the antibiotic that is your first choice? You can see that there's a lot of variation across the world, both within region, but also particularly across regions. So, for example, in North America, 78% would have chosen a cefazolin, whereas in Europe and the UK, 81% would choose an anti-staphylococcal penicillin. So we're left in this kind of area where there's a lot of variable care being provided. We don't have the answer to this very, very straightforward question as to which antibiotic we should use. And when you look at the systematic reviews, this was recently published from our group, looking at this question of cefazolin versus anti-staphylococcal penicillins, we only have retrospective observational data to look at. And in all of these, when we put them together, cefazolin is associated with lower all-cause mortality and also associated with fewer adverse effects. But of course, it's very difficult, I think, to base guideline and treatment recommendations, what we should give to patients, on observational data, which will have all its inherent biases and flaws. Have a prosthetic valve endocarditis. I mentioned that the recommendations are to add rifampin and gentamicin, and that's based on when we looked at any observational, so none of these are clinical trials on the left here. These studies are all retrospective cohorts or registries, and the size of the studies is not particularly big. And it ended up that the recommendation for this combination, when we looked back at it, I'll read this out. Therefore, it appears that the initial 1989 AHA guideline and subsequent practice of combination therapy in staphylococcal prosthetic valve endocarditis might be based on retrospective comparisons involving only 6 patients who received appropriate monotherapy without an adjunctive agent. So we're really skating on very thin ice in the recommendations that we make. And these recommendations get promulgated from guideline to guideline to guideline. For E. faecalis infective endocarditis, just to remind you again, the guidelines say ampicillin plus gentamicin or ceftriaxone. And again, when people have gone back to look at, well, what's the evidence to guide this? Well, the clinical evidence that supports this practice of combination therapy is limited to 30 patients treated with monotherapy, predominantly originating from a flawed observational 1954 study. So the problem is that registrational trials may not and typically do not address the clinically relevant questions that we have. And the follow-up trials are not happening to end up addressing these questions. And the guidelines are therefore not informed by randomized clinical trials. So my next few slides are to look at the Staph aureus clinical trials landscape. And this is the clinical trials that have been conducted in the last 20 years. We've got a year on the x-axis, the kind of phase of the study on the y-axis, and the size of the circles refers to the study size. So you can see that, right at the first one, the daptomycin trial in that blue teal color, was that registrational trial, and then the Eradicate trial to the far right was the ceftobiprole trial. And we had a couple largest trials in around 2005, a real paucity over the next 10 years, and then the last 10 years there have been an increasing number. And so these have addressed various different questions. They have been done, but clearly we need to do more studies. What you will note is that the ARREST trial, the one in the middle there, that's the largest of all these trials. So that's about 700 patients. And so in many ways, these trials are underpowered for us to really address the key clinical questions and have sufficient precision that we can have confidence in the results of them. When I put all those trials together in a forest plot using whatever primary outcome that they took and used a risk ratio, so it's a bit of a kind of dodgy forest plot in some ways, but it gives you a sense. So the red line down the middle is if there's no difference between the trial arms, and you can see that no trials have the whole of their 95% confidence interval to the left of the line, which would indicate that the intervention is better than the control. And the bottom line here is that no agent has been shown to be superior to the standard of care in Staph aureus bloodstream infection trials. And part of that is because the trials are not large enough to show that superiority. And this has then led to us not really advancing this part of the field particularly much. The mortality rate from Staph bacteremia has really sat at about 20%, and that has not changed over the last 20 or 30 years. We've never managed to find an agent that is better than standard of care. And going back to the survey, that has resulted in just no international standard of care for the treatment of this very common infection. There's a lot of variability. When you add it all up, there's been less than 4,500 patients ever randomized in Staph aureus bloodstream infection trials. And this led some editorialists, Tom Holland and Vance Fowler, to really write in The Lancet in 2017 that we just need clinical trials networks of experienced sites to prioritize the most pressing clinical questions and conduct pragmatic trials for Staph bacteremia. And I've become a very firm believer that, within a very pragmatic setting, we should be providing randomized care rather than random care and actually get to the answers to some of these questions. So how do we address that problem? I'm going to now talk about the SNAP trial. If you'd like to visit our website, there it is. You can also do the QR code. We've developed what's called an adaptive platform. The platform allows us to study a disease and not just one specific question. So we're not just studying, let's say for methicillin-susceptible Staph aureus, we're not just studying flucloxacillin versus cefazolin. We're actually studying a whole number of questions across this platform, which you can see here. We've managed to include pediatrics in this. It's been very much a community prioritized questions that we're asking. And so all these initial questions that we're addressing are ones that the clinical community are most interested in. And the platform allows great flexibility. So we're able to add in new domains as the platform progresses. And so that question of rifampin versus no rifampin for prosthetic valve endocarditis, we're adding that in soon. We're able to add in other treatments such as fosfomycin versus no fosfomycin. So as the platform matures, we'll be answering more and more questions within its infrastructure. We have core primary and secondary outcomes. So the primary outcome is all-cause mortality at 90 days. So something we really care about, patients really care about, and which is a very clear, unbiased kind of outcome. And then there'll be various other key secondary and also safety outcomes. There's really strong statistical background or backbone to all this as well. We use a Bayesian hierarchical logistic regression model. There's scheduled analyses which occur after every 500 participants. And we have really tight margins for things like non-inferiority. So the non-inferiority margin is a odds ratio of less than 1.2 for that 90-day mortality. So that if the baseline mortality is 15%, then the non-inferiority margin, the delta is only about 2.5%. So rather than a 20% non-inferiority margin, we're looking at a 2 to 3% non-inferiority margin. So this would give us much greater confidence in the non-inferiority of any of the treatments we're looking at. And the platform allows us to ask different hypotheses for each of the different cells, as I've demonstrated here. It's become quite a big study. It's a global trial, over 130 sites across 10 countries, which I've shown here. So in dark blue are the actively recruiting countries, and in the light blue are the countries and regions that we hope to be bringing on board in this next 6 to 12 months. And the funding model has been interesting that we got an initial government grant in Australia, but now countries all over the world are providing funding to support local trial activities to join the SNAP trial. So we have multiple sponsors there on the left who take responsibility for each of their regions. And on the right, you can see all the various national funding bodies which are now supporting the trial. And this is our recruitment to date from February 2022 through to March of 2025 here on the x-axis, cumulative numbers on the y-axis. The dotted lines at the bottom are the previous largest trials for staph bacteremia, and the dotted line up the top is when you add up all published staph bacteremia trials over the last 20 years, which is 4,087. So you can see that in this 3.5 years or so, we've already recruited more patients than have ever been recruited over the last 20 years. And what's more, because of the platform design, each participant can be randomized within more than one domain. So there's now more than 6,800 randomizations. This is our consort diagram up until that point. So over 13,000 screened, over 4,000 enrolled in the platform. And you can see then the numbers that have gone to each of those specific domains. And really, each of those domains now is much larger than previously conducted trials for staph in general. What we're now thinking about is, can we upscale this? This can be not just for Staph aureus, but we could have shared infrastructure which allows us to ask similar kinds of questions, but for other pathogens. So we've recently had funding to support a invasive streptococcal platform, and we're seeking funding to address an enterococcal platform as well. And if you'd like to follow for some of our recent results, which we presented at ESCMID on April the 12th, there's some QR codes for breakpoints and communicables podcast that we did recently. You can see my colleague Josh Davis there. Paul Sachs also reflected on this in his Journal Watch, and we've recently published a review article about staph bacteremia. So I'll finish there. Thank you.
Thanks very much, Stephen. Really interesting presentation. Next up, we have Patrick Harris, who is an infectious disease physician at the Royal Brisbane and Women's Hospital, a specialist in microbiology at Pathology, Queensland, and a leadership fellow at the University of Queensland Centre for Clinical Research. He leads a research group with a focus on antibiotic resistance and the use of randomized trials to optimize the management of multi-drug resistant gram-negative bacteria. And he also leads a program aiming to increase the routine application of microbial genomics to clinical practice. Just a reminder, if you have any questions, please do use the Q&A function at the top right-hand corner of your screen. Thanks very much. Over to you, Patrick.
Thanks, Holly, and hello, everybody online. Always a hard act to follow Steve's incredible accomplishments with SNAP trial that I will try and reflect on some very similar themes, but for gram-negative bacteria. And in many ways, some of the concepts and problems are very similar, but there are some subtleties about gram-negatives that unfortunately make things even more complicated and challenging. I'll just see if I can get this to move. Oh, sorry, got to click a button. Sorry, everybody, just working out the technology. Okay, it seems to be working. Go back again. All right, so look, I think we've seen a little bit about this global burden of disease study, which I think has been really, really helpful to try and get our head around exactly what is the global burden of antimicrobial resistance and where that burden is falling most heavily and which pathogens are driving much of that. Whichever way you look at it, antimicrobial resistance is playing a huge role in terms of global mortality, and that's unfortunately only set to increase in the coming decades. And currently we're looking at hundreds of thousands, if not millions, of patients dying either as a direct result of antimicrobial resistance or dying with infections that are hard to treat. And as we can see, the burden of this projected into the next decades is really going to hit hardest in, parts of the world that are already feeling these effects, particularly in sort of Southeast Asia, South America, and there are many parts of the world, for instance, Sub-Saharan Africa, we don't really have very good data, and certainly, from what we can tell, this is causing a lot of problems in these vulnerable populations. So what can we do about this? And, really one of the biggest concerns within the sort of gram-negative resistance landscape has been carbapenem resistance. So any of you who are, I guess, clinically minded will understand that carbapenem antibiotics have been an incredible suite of drugs, but often reserved for our most difficult to treat organisms, and often seen as a sort of last line antibiotic that we pull off the shelf when we're really desperate and we have a very hard to treat organism. But increasingly, we're now seeing resistance to these last line antibiotics, and frequently those strains often have very limited treatment options, or sometimes even no available treatment options, particularly in parts of the world where antibiotic supply is limited. So the group of organisms that are developing carbapenem resistance have been one of the fastest growing antimicrobial resistance concern within the gram-negative group of bacteria. And as you can see, still relatively smaller numbers than other organisms, but as I said, has been increasing much faster than other resistance threats. It's been estimated from that study that if we desperately need new drugs that can treat these gram-negative organisms, particularly these more resistant strains, and could potentially avert many millions of deaths if we can find ways to treat these more effectively. And as Steve pointed out, it's our common bacteria like Staph aureus, but in the gram-negative realm, things like E. coli, Klebsiella, Pseudomonas, Acinetobacter, these are the organisms that are most commonly associated with mortality, and unfortunately are also the organisms that are most frequently developing resistance to our antibiotics. So we have a major global challenge on our hands, but unlike gram-positives or as Steve pointed out, Staph aureus is a challenging organism, but you're relatively limited in the sort of complexity of the resistance that you might encounter. If you look at the SNAP trial, we had penicillin susceptible, methicillin susceptible, or methicillin resistant, and that's broadly the sort of most important categories of Staph aureus. But looking at gram negatives, not only if you've got multiple species or groups of species, but you have multiple potential resistance mechanisms. And you also have wildly different epidemiology across the world. So, for instance, carbapenem-resistant Acinetobacter is incredibly common in many parts of the world, as you can see in this sort of panel A here. In parts of Asia, it's unfortunately become highly endemic, particularly in hospital environments. Whereas, where I work in Australia, it actually is relatively rare and only seen usually imported from overseas. So you have to understand your local epidemiology. Certainly, if you're trying to design clinical trials, you need to have a good sense of what kind of organisms you're going to encounter and where you need to recruit. But we're increasingly seeing carbapenem resistance in common Enterobacterales like E. coli, and particularly Klebsiella in parts of the world like South Asia. And unfortunately, year on year, these become increasingly common. But it's not just the resistance that you have to consider. There's also I might just skip over that. There's also multiple potential resistance mechanisms, and we'll talk a bit more about that in a second. Sorry, I might just go back. So it's worth noting that the WHO have outlined some of they've sort of categorized, I guess, in terms of the global risk of some of these pathogens, and perhaps the ones that are setting off the highest alarm bells are things like Acinetobacter baumannii that are carbapenem resistant, the Enterobacterales, which are your common sort of gut colonizing organisms like E. coli, Klebsiella, Enterobacter, and so on, that are resistant to third generation cephalosporins, so particularly things like ESBL or AMP-C producing organisms. And then out of those are these carbapenem resistant organisms, which are often multi-drug resistant to a number of different antibiotic classes. So these are the sort of critical group, but There are several others from these high or medium risk groups that are also, common and causing significant problems. But the gram negatives, as you can see, are probably the most alarming in terms of their global burden of disease and risk of complication. Now, it's often said that the antibiotic pipeline is dry, and there's some truth to that, but really I think what people are saying is it lacks innovation. So there was a period earlier in the 20th century when there was a great flourishing of drug development, and lots of new classes of antibiotics were coming to market on a fairly sort of regular, predictable basis. And if you had resistance developing to one class of antibiotic, there was some confidence that a new one would come around the corner and the problem would be mitigated. But as you can see, in the last few decades, there really has been a real dearth of truly innovative drugs coming to market. That one blip there in the sort of 2010s is cefidoracol, but even that one could argue is, again, a variation of a cephalosporin drug, although a very novel mechanism of action. But in fact, it's not quite that desperate. So while there is a lack of true innovation in the sort of pharmaceutical pipeline, there are actually a number of drugs, as we'll see, coming onto the market. But these are mainly modifications and variations and new combinations of existing classes of antibiotics. But that's still quite encouraging, and I think there is a lot of interest in some of these new compounds and where they can fit into our armamentarium, but also, I guess, how can we really understand as clinicians how to use these drugs, as they're going to be, probably relatively in short supply, often very expensive, and I think we really need to use them wisely and judiciously, and ideally with good clinical evidence. So this table's a little bit out of date now, but it just gives an example of, I guess, how many different antibiotic compounds for gram negatives, and particularly for multi-resistant gram negatives, are coming on the horizon, and many of these have actually now made it to market or are very, very close to registration. And as you can see, probably the most flourishing, promising group of agents are these beta-lactam, beta-lactamase inhibitor combinations, with a real kind of explosion of new beta-lactamase inhibitors combined with existing beta-lactam drugs. And there's quite a lot of complexity to these, so I won't go through them in great detail, but I think this is where the sort of excitement is coming in this field in terms of how these new combination drugs are going to be available or are going to be used. And then there are some actually truly novel beta-lactams like tebipenem, cefidoracol I've mentioned, and so on, as well as some other other classes of antibiotics like aminoglycosides or tetracyclines. So it's not completely hopeless. There are drugs on the horizon, but as we shall see, these are very, very hard to generate evidence to help us understand how best to use them. Now, I said earlier that the gram-negative landscape is complicated, and one of the big complications is, as I said, not only are you dealing with lots of different species or groups of organisms, but there are multiple resistance mechanisms underlying your phenotypic resistance. And increasingly, these beta-lactamase inhibitor combinations are quite directed towards certain types of AMR genotypes. And unfortunately, I think increasingly we're going to have a better understanding of what is the underlying genetic mechanisms to really know how best to use these organisms. And even in a sophisticated laboratory, often that level of genotypic detail is not readily available. And I think we're going to need a lot of new diagnostics to really help us perhaps target these drugs very efficiently. So it's complicated, and as you can see, there really aren't any drugs that treat everything all of the time. There are some that are very good for, coliforms like ESBL or AMC producers are reasonably well covered by these new drugs, but particularly things like metallo-beta-lactamases often have limitations in terms of coverage for some of these new agents, with a few exceptions. And then particularly when you get into things like carbapenem-resistant Acinetobacter or Stenotrophomonas, even these new drugs are often struggling to be effective against these difficult to treat organisms. And if you look at the right-hand column, I've sort of tried to indicate where we have some sort of pathogen-directed trials that have been conducted in recent years to generate some evidence. And as you can see, for many of these new drugs, there is either no RCT evidence, and as we shall see, the RCTs that we do have are often, unfortunately, either not particularly helpful or horribly underpowered and very difficult to interpret. So as I said, it's a complex landscape. We really do need more trials to help understand how to treat these difficult organisms. As I said, the AMR burden is complicated. It's highly variable across the world. It's not always entirely predictable, and is constantly changing with a sort of rapid evolution of these organisms, plasmid transfer, patients traveling all over the world and acquiring these things and bringing them home with them. So it's a very dynamic state of affairs. There's also, I think, a huge combination of questions we could answer. Are there ways we could combine these drugs? How long do we use them for? What other adjunctive therapies could we think about? I think increasingly we're going to see very, very directed therapies coming through the pharmacological pipeline. So maybe drugs that only treat Acinetobacter or only target certain genotypes. When we start looking at things like monoclonal antibodies or CRISPR-based therapies or phage therapies, these are probably going to be very, very targeted. And we're going to need quite a lot of microbiological information to really know who to treat and with what agent. And as Steve pointed out, and it's certainly very true for gram-negatives as well, the sort of regulatory trials that we see that are, entirely necessary and understandable why they're done to, satisfy the FDA or the EMA to get these drugs to market, almost never really answer the kind of clinical questions that we are most in need of answering. And certainly very few of these trials enroll either the kind of patients or the kind of pathogens that we really want to know more about with these complex antimicrobial resistance profiles and where perhaps the greatest clinical need is seen. And most commonly, you'll see these registration trials which are based on a sort of a syndromic approach, so patients with UTI or pneumonia or other specific infectious syndromes, but very rarely they are directed against a particular pathogen or resistance phenotype. Now, I'm borrowing a little bit here from the gram-positive world, but it sort of makes the same point. This is the kind of situation that we often get into. Ceftaroline is an anti-MRSA cephalosporin that was originally approved for skin and soft tissue infections, but a very small number of patients in that trial actually had MRSA infection. And in fact, when it was trialed for community-acquired pneumonia, they actually had to specifically exclude patients with MRSA because the comparator drug had no MRSA activity. And this is a thing we'll see again in the gram-negative world where you're generating evidence for a condition, but with the kind of infections that you would probably never use this drug to treat. And so we're left in a situation where this new drug may come to market, but is not really used very much, and we don't really have evidence to actually know whether it will be effective against the specific kinds of infection that we might want to use it for. And there have been some pathogen-directed randomized trials. Some of them industry-sponsored, some of them more investigator-initiated. These are some of the ones that have come out of industry. But good effort on them. Well done for trying. But really, despite there's only a couple more of these trials, but they really, really struggle to recruit. So these four major investments from the pharmaceutical industry in these new drugs that are developed only actually included just over 300 patients. So really, again, you're going to struggle to really make any robust conclusions from that level of sample size. This is a sort of classic example of where things often fall over. This is the CARE trial, which was the registrational trial for plazomicin, which was a novel aminoglycoside drug that was stable to things like 16S methylases. So, potentially quite a viable option for maybe metallo-beta-lactamase producing gram-negatives and other very hard to treat organisms. But they, despite millions of investment, 15 years of effort, $1 billion invested in this drug, the trial only managed to recruit 37 patients in 68 sites, having screened over 2,000 patients. And in fact, only 17 of these patients received plazomicin. Now, that actually was enough to get FDA approval. This even got into the New England Journal of medicine as a sort of short communication, and actually plasmamycin, even on the basis of those small numbers, look reasonably promising with a potential survival benefit, but numbers too small to really make a conclusive decision on superiority. But it's an incredible shame that what was a potentially promising drug, I guess, was unable to sort of really be demonstrated in a large enough trial. And as a result of this, unfortunately, Archaeogen, who's the pharmaceutical company that backed this drug, went into bankruptcy. Sales in the market when it was finally released were pitiful, and unfortunately, all that investment went down the drain. And now this is a drug that we don't practically have access to in any meaningful way. So this is the kind of challenge. You need to do these registrational trials, but they often really struggle to answer the question that we want to answer, and then these drugs often then fail in the market, and people then more and more reluctant to invest in these potentially promising drugs. So what about investigator-initiated trials? And I guess overall, investigator trials are generally a bit more successful in terms of recruiting patients. So these are a couple of notable recent examples, recruiting patients into colistin-based trials, so one for carbapenem-resistant Acinetobacter, the Overcome trial, which is for a more diverse range of syndromes and organisms. But again, only recruiting sort of around 400 patients each. Incredible effort, but again, probably not large enough to make really conclusive decisions about treatment efficacy on relatively small numbers. So we've had a recent interest in a new drug that's now been on the market for a few years called cefiderocol, which is a very interesting drug. It's a novel cephalosporin that has more stability to a number of beta-lactamases that may break down other cephalosporins. And it's combined with a siderophore side chain, which allows it to be taken up by gram-negative bacteria in what's called a sort of Trojan horse strategy, so it can get into the periplasmic space via ion transport mechanisms where it can sort of act and have its antibiotic activity. And certainly in vitro, it's very effective against a wide variety of gram-negative pathogens, including some very, very resistant carbapenem-resistant gram-negatives, including Acinetobacter, Stenotrophomonas, other non-fermenters, and your Enterobacterales. So a very promising drug that's been on the horizon for a little while. And these are the kind of registrational trials that, as we talked about, you often have to do to get through the FDA. There's a UTI trial and a nosocomial pneumonia trial. And again, compared with a standard of care like carbapenem in both these cases, it met the primary endpoint within your non-inferiority margins. It performed pretty well and gets through its kind of efficacy and safety tick of approval. But the key problem with this is that, in fact, both of these trials specifically excluded carbapenem-resistant isolates. In fact, if you look closely, there were a few patients that slipped through the net that had carbapenem-resistant isolates, but I think there was only a handful, like 10 or so. So again, we would want to use this drug. We don't really want to use this drug for your average patient with a complex UTI, but we might want to use this drug for very resistant or carbapenem-resistant gram-negatives. But these trials, unfortunately, give us almost no data to support that decision. So following these trials, they again, great credit to Shionogi who supported this, but they undertook what's called the Credible CR trial, which was again trying to really compare cefiderocol against whatever else was available, or what's often termed best available therapy for those more tricky carbapenem-resistant gram-negatives that you might see in a more real-world situation. So this was an open-label, multi-center, pathogen-directed trial, recruiting in parts of the world where they have high burden of gram-negative resistance. And the really depressing thing is actually, unfortunately, when you look at the raw numbers, in fact, there was a higher mortality in the cefiderocol arm, by quite a wide margin, so almost double the rate of mortality, so 18 versus 34%. And this was almost entirely driven by patients with Acinetobacter infection. So this, unfortunately, put a fairly dark cloud I go over copheterocol's use, particularly in patients with difficult Acinetobacter infections. And this also highlights the real challenges of sort of when you look at these drugs in real world situations. And there's been a lot of debate about what happened with this trial. Certainly there probably was a bit of overrepresentation of patients in the copheterocol arm getting Acinetobacter who were in ICU or had renal failure or had other potentially confounding conditions. So people have tried to explain it away, but it's certainly been a worry for clinical use. So through our centre here at the University of Queensland and with our colleagues over the world, or across the world, who have much more resistance than we do, we try to, I guess, really tackle this question of, what is the utility of copheterocol in the real world, and how can we use this drug in parts or in settings where you have a high rate of gram-negative resistance. We had an interesting model where the drug was supplied by Shionogi and they provided some of the funding, but it was sponsored by the University of Queensland, who had control over the running of the trial. So we had autonomy to run the trial as we thought was most in the interests of answering the clinical questions we wanted to address. So good on Shionogi for taking a bit of a risk and allowing us to test their drug out. By which point it had already made it to market, which makes life a bit easier for them. So we recruited in parts of the world again with high rates of resistance, places like Singapore, Taiwan, Thailand, Turkey, and so on. And after, again, these trials are very, very difficult to do, but we managed to get just over 500 patients, of which about a quarter had carbapenem-resistant gram-negative bacilli. So these were healthcare-associated gram-negative bacteremia patients. So we recruited them at the point at which we knew they had a bloodstream infection with a gram-negative. And we enriched for resistance, I guess, by recruiting only healthcare-associated. But the great challenge of these sort of trials is you very often don't have information about carbapenem resistance up front, particularly in parts of the world where rapid diagnostics are not readily available. So we slightly had to enroll blindly, and we hope to have got more carbapenem-resistant gram-negatives, but as is always the case in these sort of trials, you never quite works out in the way one would hope. But I think 127 patients is probably not too bad, but again highlights the real challenge of getting big numbers into these kind of trials. We had a primary outcome of all-cause mortality at 14 days, and we compared copheterocol to a standard of care. And again, this is very hard to define, and we'll talk a bit more in a minute about the challenges of defining that standard of care where there is no real standard of care. But we could say at least that in our primary outcome, there was essentially no difference between copheterocol and standard of care. So for your undifferentiated gram-negative bacteremia in an area of high resistance, it is certainly as good as standard as care. But it met non-inferiority, but it didn't really achieve superiority. And I guess one might question, well, if you're really going to use this new expensive drug against what are often not very brilliant standards of care like colistin or other relatively old-fashioned drugs, should you really hope for a greater effect than this? And I think perhaps we didn't really see much difference between standard of care and the intervention was, I think, relatively disappointing. And this is probably even more important. Oh, sorry, I should mention at least one good news we found was that in fact we didn't replicate that concern over Acinetobacter. And in fact, patients seem to do better on copheterocol with Acinetobacter infection. So I don't know if that's eliminated that issue, but it was good to see that we didn't see that replicated from the credible trial. But I think some of the most disappointing results for us came out in the carbapenem-resistant subgroup. So these are patients who are infected with a carbapenem-resistant gram-negative. Again, not big numbers, 60 odd in each group. But again, numerically higher mortality in copheterocol. Now, these numbers are too small to have much statistical inference from that. It's underpowered. But the fact that you're not seeing superiority, if anything, somewhat the opposite, is, I think, a disappointment. For a drug that should be performing much better if you really wanted to use it. But these are very sick patients. You can see that 50% of them are dead by 90 days. They have many other comorbidities and many other things going on. But to me, one of the greatest concerns is actually we've been looking at the resistance of these organisms, and particularly those that have acquired metallo-beta-lactamases, particularly things like NDM, so New Delhi metallo-beta-lactamase. Almost 60% of these strains are already resistant to clofidogrel when you test them by a reference broth method. And unfortunately, there's a whole emerging story about resistance to clofidogrel, particularly in NDMs and other challenging organisms, that I think unfortunately is going to compromise this drug before it's even had a chance to really get out into the real world. So I can't pretend that this has answered all the questions, but I think these are exactly the kind of trials we need to do on a grander scale to start tackling these really important questions, rather than just assuming these drugs are going to be fantastic because they work in the test tube. We really need to understand how they perform in the real world with patients with these kind of infections. I'll also mention some other new drugs that are sort of coming on the horizon. So many of you may know about a drug called aztreonam-avibactam, which has now reached approvals and is available for clinical use in some parts of the world. One of the big problems in treating carbapenem-resistant Enterobacterales are these metallo-beta-lactamases like NDM producers. Often there have not been good drugs to treat these metallo enzymes. And this quite clever manipulation of using aztreonam, which is actually an old-fashioned drug but is stable to metallo-beta-lactamases, with this novel beta-lactamase inhibitor avibactam, which is normally combined with ceftazidime, but combined together in this new formulation, the avibactam can knock out your class A, C, and D enzymes and leave the aztreonam to be active against the class B metallo-beta-lactamases. And certainly in vitro, this looked like a very good drug, and obviously this kind of thing we would want to start potentially using against particularly these class B producers. And the best trial we have has recently been published just this year, called the Revisit trial, which was, again, a slightly odd. They described it as a descriptive RCT. I didn't know one could have a descriptive RCT, but essentially that means it's a randomized trial without formal hypothesis testing, which essentially means it's underpowered as usual. But this was enough to get it over the line in terms of efficacy and safety. It works. It does what it says on, one would hope to, on the tin. But when you really delve into the details of a trial like this, as I said, this is a drug that we would really want to use against metallo-beta-lactamase producing organisms. Only 10 patients in this trial had those kind of infections. So again, unfortunately, it really doesn't tell us much about what we can do when we're faced with a patient with these kind of difficult to treat organisms. This is probably a slightly more encouraging result, and a rare thing you see in gram-negative trials, where you actually see a fairly substantial mortality benefit. So this was the ATTACK trial, which was looking at a novel drug called sulbactam, durlobactam. In fact, important to note that it's always combined with imipenem in this trial, but compared to drugs like colistin for carbapenem-resistant Acinetobacter. Again, an incredible effort, 16 countries, 60 sites, still only recruiting 125 patients actually with a crab infection, but again, one of the largest crab trials, or certainly from a sort of industry-sponsored trial, and in fact was able to show quite a substantial mortality benefit, not quite reaching full statistical significance, but as you can see from the forest plot there, a reasonably impressive, consistent favoring of sulbactam, durlobactam, but with relatively wide confidence intervals, hence not quite all to the left of zero. But probably the real kicker for the drug like this is, the much lower incidence of nephrotoxicity, because you don't have to use colistin. So this is a kind of trial that I think is quite convincing. It sort of tells us that you're probably going to be seeing a mortality benefit, which is ultimately what we want to see, and much lower toxicity. But the problem with a drug like this is, I think in our hands, it's about $5,000 a day. So it's really going to be beyond the availability of many parts of the world that actually see patients with crab infection. So again, highlighting the kind of odd asymmetry of, the sort of inverse care law of antibiotics, unfortunately. So what can we do about this? We've got registration trials that don't really answer the questions we want to see. Plucky investigators try and do these kind of studies, but we're really hampered by the limitations of what we can achieve. We're often underfunded. We're often doing multiple other things. So to do many thousand patient patient enrolled trials to get that kind of adequate sample size is incredibly challenging. I think Steve previously really has shown the light in terms of how this can be done with the SNAP trial. And I guess the question is, can we do a similar thing with gram negatives? Is that a success we can replicate? But as we've sort of seen, the landscape is so complicated because it isn't just one organism. We're looking at potentially multiple different organisms, multiple different resistance patterns and AMR genotypes. So how can we do this? There isn't really a standard of care. Everybody's doing all kinds of different things. How can we design a trial that could work? And this is actually a really nice design that's come out of the UK from Sarah Walker, who some of you may know about, who's a very, very eminent clinical trialist. But as I said, the difficulty is what do you do when there's no standard of care? What do you do when no one really knows what the right there's no established treatment that you can compare your new treatment to? We've often used this thing called BAT, or best available therapy. They're pragmatic and they're, I guess, easier to sort of allow into your trial, but they're incredibly heterogeneous, and every hospital's doing different things, every clinician's doing different things, the dosing's different, the combinations are different, and then it becomes really, really hard to understand what your comparator is doing. The other challenge is if you're really trying to recruit real-world patients, is they're complicated. They've got renal impairment, they've got all kinds of comorbidities, they're often maybe elderly or frail. There may be challenges in terms of drug availability. There may be that in your country some drugs are just not available, or they're incredibly expensive, or they're not approved for use, or they require special, regulatory shenanigans to get them to the patient. And so this is a design called the practical design that tries to, I guess, address some of these complexities that are particularly prevalent in treating things like multi-resistant gram negatives. And the idea is that you essentially generate a personalized randomization list that you are all agreed upon in your institution, for CRE or CRAB. We are happy to use these 3 or 4 drug options. That might be colistin with high dose meropenem, ceftazidime-avibactam if we have a KPC infection, for instance, or an OXA48, ceftazidime-avibactam plus aztreonam for metallo-beta-lactamase. Whatever you are agreed upon in your institution that would be a suitable option or an available option. And then you can essentially generate your list of your personal randomization list. Hang on, sorry, my slides are not moving. See if I can do that again. There we go. And let's say, for instance, in this situation, you have an elderly man with acute kidney injury. They've got a metallo-beta-lactamase producing enterobacter in their blood. They've got a reasonably high meropenem MIC, but not, it might still be in the intermediate range. And let's say you can't get IV fosfomycin locally in your hospital. So as a result of some of those restrictions, you look at your randomization list of available options. And given some of those restrictions, well, you don't want to give plazomicin or colistin, perhaps because of his renal injury. Let's say you don't have some of these drugs are not available, so you can't use intravenous fosfomycin, but you can randomize into cefirocol, high-dose meropenem, double carbapenem treatment of some description, and so you have some options available to you that you could treat in this situation, and you randomize to those available options. And then throughout your entire trial network, you do essentially like a network meta-analysis of all those different drug options. And hopefully, if you recruit enough patients, you can have enough comparisons to ultimately make essentially rank which of these options are most effective, and ultimately drop those that are performing the worst and add in potentially new questions as you go along. So this is a kind of trial design that we're playing around with to try and build a gram-negative platform, and it's already underway in Singapore, but we're hoping that we can have more parts of the world joining in coming years. But the real challenge, I think, is going to be identifying these patients really early, because we're going to need to know what kind of AMR gene they have. Have they got an NDM? Have they got a KPC? I think more and more we're going to need things like rapid diagnostics to bring these patients into these trials, and this is a trial based on that concept that's now recruiting in Southeast Asia, which I think are going to be very interesting to see how they can use things like rapid molecular testing to enrich enrollment into these studies. So I think I'm out of time, and I think that's my last slide, so I will stop there. I think I've kind of made my points, and I will hand it back to see if there's any questions, and thank you for your attention.
Thanks very much, Patrick. Wonderful presentation. Thanks very much to both of you. Don't forget, if you're listening, please feel free to ask questions using the Q&A function at the top right-hand corner of your screen. So let's dive straight in. Stephen, first question for you. So many trials of newer agents don't offer contemporaneous antimicrobial susceptibility testing. Did the trials that you presented on do AST as or prior to enrollment?
Yes, absolutely. So the entry into what we've called different silos actually depended on what the susceptibility profile was, be it penicillin susceptible, methicillin susceptible, and penicillin resistant, or methicillin resistant. And for the distinguishing of whether it was penicillin susceptible, in fact, we required more than just the automated antimicrobial susceptibility testing. We had sites had to do a disc phenotype as well to confirm penicillin susceptibility.
Great.
But
thanks.
I think, as Patrick says, it's much easier, I think, with Staph aureus than is with the gram negatives. There's there's BLAZ or MEC-A. They're the two genes which are involved.
Yeah, leading on from that, Patrick, you highlighted this as a potential issue. Pathogen-directed trials are potentially becoming a bit more common. Will the diagnostics be able to keep up? Can these trials be done in parallel, or do we need to focus on one or the other maybe first?
Yeah, no, it's a great question. It is a big challenge. I think the trade-off is, even rapid diagnostics that are sort of generally available in most labs are still relatively slow. We have MALDI-TOF, and we have, RAST early susceptibility testing. So we have things that can give you a reasonably early clue, but it still might be, 12, 24, 36, maybe up to 72 hours before you really get the information you want, plus all the other delays you have with just enrolling patients into trials. And that's the eternal tension. That's the thing we found in Game Changer. We had a pretty tight limit in terms of when we could enroll, just because we wanted to, we didn't want to enroll people at day 5 because they've had probably most of their effective therapy by that point, and you're just tailing with the edges. But you don't often have that information, within the first 12 hours of them flagging with an infection in terms of the susceptibility. Now, I think I mentioned right at the end, I've probably glossed over it a bit, but this rapid trial that's using BioFire to do sort of upfront molecular diagnostic to tell you, yes, you have an Acinetobacter and there is an OXA23 carbapenemase. Therefore, you can be enrolled into this arm, or whatever, or something else for another drug. I think that's going to be the kind of thing we really need, but that's very challenging. It's very expensive. It needs a really highly functioning lab that knows what it's doing. It needs lots of communication between the clinical and microbiological arms of your study. So it's a real, it's not easy to do. I think actually that rapid trial is recruiting pretty well. I think particularly from India and places like that. So I think it can be done, and it's a real credit to some of these places that probably have not been particularly prominent in clinical trial world, maybe coming up to the plate and actually really beginning to tackle some of these questions in the parts of the world where it's most important. And that's been the big problem is, the places that have the money and the resources and the experience and the teams of people to do it don't have the resistance. So we can't recruit enough patients, or we try and we just never really get to the numbers that we'd like to see. So I think doing these studies where the problem is going to be the key at the end of the day. But they're going to need a lot of help and support to get the lab diagnostics up to speed and to do it properly.
Yeah, that links in quite nicely to the next question for Stephen. Given that most of the countries involved in the SNAP trial are high-income countries, are there plans or steps being considered to include more low or middle-income countries in future phases or related initiatives to enhance equity and inclusivity in the trial's findings?
Yeah, what a great question, and very, very pertinent. We're trying, and we've tried. It's actually not straightforward. So we have South Africa as one of the sites, and in fact, we're trying to write a manuscript at the moment trying to describe the challenges of even bringing something like SNAP. So it is tricky because as part of SNAP, we do need to know if it's MRSA or MSSA or PSSA. And it might sound simple, but the lab's not always able to do that in a timely fashion, much less these complicated gram-negative resistance profiles. So really simple things like even, there'll be whole weeks when they don't have the necessary plates to be able to plate out the bacteria to do the specific susceptibility testing. And so it's been interesting. When we designed SNAP, we didn't really have low middle income country areas in mind. And moving forward for STRAP, which is the streptococcal one, we've really made a conscious decision to say, actually, we've got to get all these regions or people from low middle income country regions on board early on and engaged, and actually help us in designing the trial, so that it's not, us foisting a trial protocol into somewhere where the trial protocol is not going to work. We've got to actually work with investigators from these regions to make sure that from the original design, it is potentially fit for purpose for those areas as well.
Yeah, I think the conversations are really important to be having. If you're trying to include these countries, then you need to make sure your protocol is valid in those countries, definitely. Thank you. Patrick, again, along a similar line, what policy or strategy should be used to ensure access to new antibiotics in resource-limited countries, especially given that antibiotic use is not well controlled in this context? And is there a risk of resistance emerging to these new antibiotics?
Again, excellent questions, and I wish I had an easy answer. I mean, the fundamental problem is, at the moment, this is very much left to the market, and these companies have invested millions and millions into these drugs, and they're going to recoup their investment in wealthy countries ultimately. And even in Australia, we're not often a target for their early sort of marketing because we're such a small population and so on, things like that. So certainly, lower middle income countries are very, very far down the sort of attention of the pharmaceutical industry. For understandable reasons, they're businesses and they're there to make money, unfortunately. But I think this is where other institutions really need to come to the plate and think about innovative ways to deliver these drugs to where they're most needed. I mean, credit to GARDP. So GARDP has a quite impressive plan to sort of deliver cafetericol to, I think, 130 odd countries through sort of a generic supplier. I think things like that are going to be really, really important. But as the questioner says, it's going to be critical that these drugs are used properly. If we burn through our most precious resources, we're going to be in an even worse situation. I fear already there are big problems with cafetericol. I know Shionogi are desperately working to try and combine cafetericol with new inhibitors to try and mitigate some of these resistant issues that are already emerging. There's a big problem with PBP3 insertions with NDM, E. coli, and so on, which are really becoming quite common, and we certainly saw that in our trial. So I think it's like everything, we definitely have to find ways to deliver these drugs to where they're most needed, but it's going to have to be done in a very thoughtful, controlled way, so it's not just a free-for-all, and we burn our bridges before we've even tried to cross them.
Yeah. On the topic of sulfuricol, this person has said the findings are ugly indeed, and you say it casts a cloud on the drug usability, but you are not mentioning the potential of combining it with MBL-specific BLI that would compensate completely the effect, maybe?
Yeah, no, that's kind of what I was just alluding to then. And look, I think sulfuricol is still a very good drug. I think the problem is that there's more resistance than we realized. I think again the problem was a lot of those in vitro studies were probably done in samples from North America or Europe, and we didn't really probably sample the parts of the world where the resistance is at its worst. And it's when you start to use this drug, or you look for the sort of what's happened. I guess also we're sort of reflecting some of those studies were samples that were probably collected 20 years ago, and things change very, very quickly. So yes, I think, as I said, Shionogi are looking very hard to combine cafidrocol. They've purchased QPEX, which has this new inhibitor called zerubbabactam, which is incredibly broad spectrum inhibitor that essentially is sort of a universal inhibitor for all beta-lactamases, which is quite promising. Some of the resistance, though, to cafidrocol is not all beta-lactamase driven. If you have PBP mutations, it doesn't probably matter what inhibitor, well, it may help a bit, but the inhibitor is not going to overcome those PBP mutations. So it's, once again, unfortunately, no free lunch, no easy answer, and just highlights the need to just be super careful about how we use these drugs. Don't waste it on trying to treat in bacteria that are already resistant, and that sort of thing will just breed more and more resistance. So I hope that there are ways to, and again, the problem is even if they come up with a new inhibitor combination, it could be many, many years before it's actually fully viable and available. So it's a big challenge.
Yeah, indeed. I think we'll move on to the last question now. I think we're almost out of time. Stephen, this one's for you, and I'm sure you could spend an hour talking about the answer to this question. This person was very upset you stopped when you did because they wanted to hear more about the results from the SNAP trial, as I think everyone does. Is it too early to conclude on anything, and what can one actually expect to get out of the platform trial?
Yeah, look, we have presented the results publicly at ESCMID about a month ago now, so I can say something. With MSSA, we recruited about 1,341 patients in that comparison between cefazolin and flucloxacillin, and we found that, sorry, it's just my mother calling, so I'll tell to call later. We found that, in fact, we met a non-inferiority trigger for cefazolin versus fluclox. So cefazolin mortality was 15% and flucloxacillin was 17%. So we had a 99% posterior probability that cefazolin was non-inferior, and about a 90% posterior probability that it was in fact superior on that primary outcome on 90-day mortality. In addition, cefazolin caused less acute kidney injury, and we were very confident in that, in the superiority of cefazolin for acute kidney injury was over 99% posterior probability. So both, I'm pretty convinced that cefazolin is both more effective at that mortality level and also safer as a drug to use, and it's led to me changing my practice. So I'll leave that there. If you want to hear more about it, do go to that Communicables and Breakpoints podcast, where we discuss the results over about a 1.5 hour period. So if you want the details, go there.
Yeah, I can really recommend the podcast. I was listening to it the other day. Really, really interesting results. I might just ask one more follow-up question on that. I'm really interested in the stopping rules that you have within SNAP, and please correct me if I'm wrong, but they're different per domain. Some are testing superiority, some are non-inferiority. For the ones that find non-inferiority, would you then go for a superiority test? Would you keep on doing the trial to try and find that superiority conclusion, or not necessarily?
Yeah, so it does depend on which part of the trial or which domain. And in fact, for example, the cefazolin versus flucloxacillin, our statistical plan was that if it met non-inferiority, the Data and Safety Monitoring Committee could let it go towards superiority to meet the trigger for superiority. Now, they decided not to do that because of the toxicity, but they could have done so. And seeing the data in the background, the rest of the trial infrastructure and investigators would remain blinded. So we wouldn't know that it had hit non-inferiority. We would just be told to keep going until potentially it hits superiority as well. But of course, that's not quite what happened.
Yeah, and that's due to the toxicity in the standard of care arm?
Yeah, controlling the flucloxacillin arm.
Yeah. Okay, great. Thanks very much. Okay, thanks very much for your presentations, guys. Thanks very much for everyone's questions at home. Shireen, back to you.
Thank you so much, Holly, for being such a great moderator for our discussion today. And of course, thank you also, Stephen, Patrick, really interesting talks. Thank you to our audience. There were so many questions. We unfortunately couldn't go through all of them, but a very, very interesting topic. So thanks to everyone. So our next Revive webinar will be announced soon. You can keep the news channels on the lookout for that. And I also just wanted to say a quick word about a conference that GARDP is co-organizing. That's the Global AMR Innovators Conference in London, October. So for this conference, there will be 10 travel grants for early career researchers and also 10 for residents of low or middle income countries. So applications are open on the website until the 2nd of June. So once again, thank you everyone. For the audience joining as well, if you just stay online, there's going to be a survey coming up. We would really like to hear more about what you thought of this presentation, or if you have any comments about our further ones. And thanks for tuning in. Have a good day, everyone. Goodbye.
Thanks very much.
Bye.